Our lab is interested in the elimination of complex cells, which have long projections spanning different microenvironments, and different parts of these cells will die differently. We study this in an organism called C. elegans, which is a roundworm that's transparent, allowing us to see the cells dying live, and we're able to take images and movies of these. It's also really easy to work with and has an invariant lineage, meaning that the cells will die the same way at the same time in every generation, and we can repeat a lot of experiments. Uh, my objective is to understand the link between complex cell elimination under normal development and stress. And thus, I subject my animals to heat stress prior to looking for defects. So our system here is a novel death program called compartmentalized cell elimination, or CCE. It's characterized by a cell dying and forming three distinct compartments as it dies. Here we have a cell that's responsible for forming the tail of the worm prior to dying in the embryo. And the cell body will start to round up as it dies. The proximal end of the process will bead, and the distal end of the process will retract into itself, leaving us with two fragments that are engulfed by two different neighboring phagocytes. So I look at mutants for sequest1, which is a selective autophagy receptor, which is a process of cellular quality control, which we recycle damaged or misfolded components and proteins of the cell, recycle them to use in different parts of the cell without killing the cell. And under normal growth conditions at 20 Celsius, I don't see a defect in CCE, which we measure by persistence of the cell and cell fragments inappropriately. But following heat stress conditions, I do start to see persisting cells. And its expression pattern shows the sequest1 is expressed in the neighboring cell, which is the phagocyte. And based on cell-specific rescue experiments, we see cell non-autonomous function. These mutant remnants also appear to be already internalized by the neighboring phagocyte. So suggesting this function of sequest1, or rather the defect, is happening somewhere in following the internalization step. Next, I look at skin1 mutants, which are promoted by sequest1 mutants, and they show a similar defect, nothing under normal conditions, but the effect of CCE following times of stress. They also show a similar expression pattern, also show cell non-autonomous function, and they also appear to be internalized, suggesting, again, defect in the later steps of phagosome maturation. Skin1 is a transcription factor. So one of its potential targets is known as list1, which regulates lysosomal trafficking. So we looked at list1 mutants, and again, we see a similar CCE defect as we do in our other two mutants, only following times of stress. It also appears list1 has cell non-autonomous function. And list1 only seems to be expressed in the neighboring phagocyte following conditions of stress. And we look at this expression marker in sequest1 and skin1 mutants, we see that expression is decreased significantly, suggesting that list1 only needs to be transcribed in times of stress to help aid in corpse digestion. Furthermore, skin1 typically is known to translocate to the nucleus and be activated in times of oxidative stress. So we just wanted to test if the same thing was happening under heat stress. So we look at a skin1 GFP marker, and we do see translocation to the nucleus in our heat stress conditions. Next, a focus on lysosomes led us to look at LAMP1 mutants, which is an important lysosome membrane protein, and they show similar CCE defect as well, and they also seem to have cell non-autonomous function. Lastly, when we mechanisms of autophagy and phagocytosis, the question of LC3-associated phagocytosis, or LAP, always comes up. LAP is a type of non-canonical phagocytosis, or cell clearing, that uses parts of the autophagy machinery in which our sequest1 mutants are involved, but is not actually autophagy. And it's regulated by a protein called Rubicon. So we looked at mutants for Rubicon, and we see similar defects again to our other mutants. So our proposed model here is P62, by inhibiting skin1's inhibitor, promotes it, allowing it to translocate to the nucleus, transcribe its target, potentially list1 in this case, to increase the trafficking of lysosomes to the cell corpses, allowing them to be digested and cleared in times of stress with potential assistance from Rubicon and the LAP machinery. It's significant because this is a new role for skin1 not previously shown. It also is potentially a new context for LAP and C. elegans. And my next step mainly is to overexpress skin1's inhibitor and see if I see a similar defect. Also further identify the role of Rubicon in this context. And most importantly, to further confirm that skin1 actually is transcribing list1 in this context,